Again, good morning and welcome, my dear medical students, neurophysicians, neurosurgeons, and neuroscientists. In my last lecture, I was talking about the structure of the neuron and axon. So, this is lecture on general neuroanatomy. In my last lecture, I was talking about the structure of the neuron. It is having, a typical neuron is having multiple dendrites, arborization of the dendrites, that is the branching and rebranching of the dendrites, forming a tree-like structure, and a long single neurite or process known as axon, and it is having branching on its right angle. In the side of dendrite there is tapering, but there is no tapering of the axon, and the most distal branches are in form of telodendria, and it is having a myelin sheath. Myelination in CNS is done by oligodendrocytes or oligodendroglia. In case of PNS, it is done by Schwann cells. So this is insulator sheath formed by the glial cells. Now, the collection of these axons is known as peripheral nerve in PNS and tract in CNS. So, what is a peripheral nerve? And what is a tract? Here is collection of neurons, that is collection of perikaryon or soma, and these are their axons coming out from the soma or cell body of neuron. Here is other neuron, one more neuron here, and these are remaining neurons, axons. So this collection of the soma or the cell bodies inside the CNS if this collection is inside the CNS, known as nuclei. Nucleus or nuclei. When this collection of cells is seen in peripheral nervous system. This collection is known as the ganglia or ganglion. The axons arising from the same nuclei or same ganglion. If these are arising from the central nervous system and running inside the central nervous system, this collection of axons is known as tract. But when this collection of axons leave the central nervous system and enters in the peripheral nervous system, this collection is known as peripheral nerve. But there is a difference in the tract and peripheral nerve. The tract is having same fibers, that is fibers taking origin from the same nuclei and terminating in the same nuclei or same part of the brain or CNS. But in case of peripheral nervous system, this collection of axons is known as peripheral nervous system, but it is having different fibers from different nuclei and ganglion. For example, in spinal cord, when we see the transverse section of the spinal cord, we will see multiple tracts or collections of axons. Here is section of spinal cord having posterior median sulcus and anterior median fissure. Here is dorsal column. Here is dorsal horn, lateral horn, anterior horn or ventral horn, again lateral, and this is dorsal horn. Here is central canal of the spinal cord, and here are different fasciculus or funiculi. This is dorsal funiculi having fasciculus gracilis and fasciculus cuneatus, and here are anterior and lateral spinothalamic tract, spinocerebellar tract, and spino-olivary, spino-vestibular, spino-tactile, spino-reticular, and some descending tracts like corticospinal tract, rubrospinal, and reticulospine, tactospinal tracts. So these bundles of axons inside the central nervous system running vertically upward and downwards are known as tracts. But when we see the peripheral nerve is having multiple origins and different types of fibers. I will draw a spinal nerve. Spinal nerves taking origin from spinal cord, again posterior median sulcus, anterior median fissure. Here is dorsal horn, lateral horn, anterior horn, and again dorsal horn of gray matter. Here is central canal. This is sensory root. And here is the motor or ventral root. Here is the sympathetic ganglia and sympathetic trunk. Running on both sides of the vertebral canal. And here is the dorsal ramus. This is dorsal root and this is dorsal ramus. So this is a typical spinal nerve having a dorsal root, ventral root. Dorsal root is pure sensory, ventral root is pure motor, and here is the trunk of the spinal nerve, and it is having connection with the sympathetic chain and sympathetic ganglion. The fibers which are coming from the motor horn, fibers from lateral horn will enter inside the lateral horn, enter inside this ganglion. These fibers which are entering inside this ganglion are known as the white ramus, and fibers leaving this ganglion having postganglionic fibers re-entering inside this nerve and supplying the smooth muscles. These are smooth muscles. These are postganglionic autonomic fibers supplying the smooth muscles of viscera, supplying the heart, and supplying the glands. Smooth muscles, glands, and heart. So these fibers which are preganglionic known as white ramus communicant, and fibers which are postganglionic known as grey ramus. These are unmyelinated fibers, so these are grey matter or grey ramus communicant. The other types of fibers from motor grey horn are the somatic efferent, without relay in the sympathetic chain. These fibers will supply the skeletal muscles. Skeletal muscles. The fibers taking origin from the motor or ventral horn and relay in the sympathetic ganglion, postganglionic fiber arises from this sympathetic ganglion, will supply the smooth muscles, the cardiac muscles, blood vessels, muscles of blood vessels and glands. Fibers from these nuclei situated in the ventral horn of the gray matter will supply the skeletal muscles, tendons and joints. The fibers which are entering in the peripheral nerve from the skin and from the viscera, that is blood vessels, heart and glands. These fibers, all sensory fibers, either they may be somatic or visceral, are having their soma inside the dorsal root ganglion. This dorsal root ganglion is having the pseudo-unipolar or unipolar cells which are having a peripheral process which is bringing sensation from the periphery, that is skin, muscle, joints, tendons, viscera, blood vessels, glands and viscera of the thorax, that is lungs and heart. The central process of these cells will enter inside the sensory gray horn or dorsal gray horn. These cells are having their second order neuron in the dorsal root ganglion. First order neurons are in the dorsal root ganglion and second order neurons are in the dorsal horn of the spinal cord, but some fibers are having second order neuron in the brainstem, that is medulla. The nucleus gracilis and nucleus cuneatus are having second order neuron in the spinal cord in the medulla, that is brainstem. So this is a typical spinal nerve having four types of fibers. One is somatic efferent supplying the skeletal muscles, joints and tendons. The visceral efferent taking origin from lateral column or lateral horn of the spinal gray matter, and fibers will enter inside the sympathetic chain and sympathetic ganglion, and after forming synapse, these fibers which are postganglionic now will re-enter in the spinal nerve and will supply the smooth muscles of the viscera, glands, blood vessels and heart. Third types of fibers which are entering inside the peripheral nerve are somatic sensory. These fibers are having cell body inside the dorsal root ganglion of the spinal cord, and viscerosensory fiber coming from the heart, viscera, blood vessels, glands will also bringing sensation towards the dorsal root ganglion and central process entering inside the dorsal horn of the spinal cord. So this is typical spinal nerve. Some fibers which leave the sympathetic chain without relay inside will form subplanchnic nerves, greater, lesser, least subplanchnic nerves, which are also having preganglionic fibers. These nerves will relay in the prevertebral ganglion to supply the viscera of the abdomen. So axon is having myelin sheath, formed by oligodendrocytes inside the CNS and Schwann cells outside the CNS, that is peripheral nervous system. There are some diseases which destroy this myelin sheath and will lead to serious neuropathies. If this myelin sheath is damaged by autoimmune disease in peripheral nervous system, this is known as Guillain-Barré syndrome. Guillain-Barré syndrome, and this myelin sheath is destroyed by autoimmune antibodies inside the central nervous system. This condition is known as multiple sclerosis. Where optic nerve is also affected, because optic nerve is not a true nerve. So myelination of optic nerve is done by oligodendrocytes. So in multiple sclerosis, optic nerve is also damaged, and some other diseases which affect this myelin sheath, like Tay-Sachs disease. In this disease, GM2 gangliosides will deposit in the myelin sheath and will affect the conduction of the impulse in the axon. Now, what is action potential? What is action potential? This is an electrical impulse. When we receive some sensation from the outside or inside the body, or from the proprioceptive receptors, these receptors will convert this sensation in the electrical impulse known as action potential. So action potential is single transient reversal of polarity of the membrane. So what is action potential? It is single reversal transient single reversal transient change in the polarity of the membrane. Normally, resting membrane potential, the inside of the axon is having minus 50 to minus 70 millivolt of electrical current, and it is having sodium ions outside and potassium ion, concentration of potassium is inside. When this terminal part of the axon, which is converted in receptor in case of sensory neuron, when someone touch or there is pinprick or burning or sensation of heat, these receptors will convert this mechanical or chemical or photo sensation in action potential. So receptors are working as transducer, which change one form of the energy in the other form of the energy. This changed form of energy, now known as action potential or the impulse, this will travel from the one node of Ranvier. In my last lecture, I have told what is node of Ranvier and what is myelin sheath. So, change in polarity is seen in only node of Ranvier. There is no change in polarity. So, conduction is very fast. So, axons can propagate the impulse from one end of the axon to the other end, that is for very long distance, unattenuated form. The amplitude and duration of the impulse is not changed. So there is jumping of action potential from one node of Ranvier to the other node of Ranvier. This type of conduction is known as saltatory conduction. And the speed of conduction in fast conduction may be many times more in comparison to the unmyelinated fibers. The speed of conduction depends on two factors: conduction speed, one is diameter of the axon and other is the myelination status. For example, the fasciculus cuneatus and fasciculus gracilis are heavily myelinated thick fibers and having very rapid conduction of the impulse from lowermost part of the spinal cord to the uppermost part of the brain. So speed of conduction depends on diameter and myelination status of the axon. So what is axon and what is conduction? We have seen. Then what is the receptor? Receptor is a transducer, biological transducer, which convert the sensation, maybe due to heat, due to cold, due to some injury, pain, or in form of hearing and the vision. These sensations will be converted in a mechanical, in a electrical energy, that is action potential. So this biological transducer is known as receptor, and the energy which is changed from mechanical or chemo to the electrical is known as impulse or action potential. Now, after a short break, I will continue with the types of receptors and what is blood-brain barrier and what are the meninges and what is CSF and what is ventricular system of the central nervous system.